Inside every living cell, a miracle of biology unfolds, the transformation of genetic code into functional protein. It all begins in the nucleus. DNA is transcribed into mRNA, a single-stranded messenger that carries genetic instructions out into the cytoplasm. Once in the cytoplasm, the mRNA meets a ribosome, the molecular machine built from large and small subunits. This marks the start of translation. As the ribosome reads the mRNA codons, tRNA molecules deliver the corresponding amino acids. Each codon matches with a specific anticodon on tRNA, ensuring the correct sequence is built. Like a protein factory line, amino acids are linked one by one, forming a growing polypeptide chain, the primary structure of a protein. When a subcodon is reached, translation halts, and the new protein is released. But the story does not end there. Proteins undergo post-translational modification. These modifications transform a simple chain into a functional protein, an enzyme, hormone, or a structural unit ready to do its job. From the genetic code to the cellular function, this is how life builds itself, all in a day's work inside the cells.